Good morning once more. Welcome to this general microbiology lecture. Today we are going to be talking about viruses. We'll be looking at how to isolate viruses, how to culture viruses, and how to identify viruses after our culture. So there's a lot to do about viruses, but unfortunately, I have just a single lecture for that. So I will try to be very, very succinct and ensure that you grab the basics of viruses. As you already know, viruses are considered to be cellular particles. Viruses do not have the capacity for independent cellular metabolism. They will need another cell to be able to synthesize proteins. They will need a cell to be able to even replicate themselves. So that makes it very, very difficult and complex to deal with. And because of this, we are unable to culture viruses in the laboratory using the normal, typical synthetic media that we use for bacteria and parasites. We need living media, whether human tissues, animal tissues, or even bacterial cells to cultivate viruses in the laboratory. So I have different lectures I'm going to be using to bring this concept as close as possible to you. The first thing I want to also appreciate is the viral particle. A viral particle is a simple nucleic acid material enclosed into a protein coat. You have a nucleic acid material enclosed into a protein coat. That is typically what a viral particle is like. It is the smallest infectious organism. Some nuclear material range from as little as 4 genes to 100. So you might have a virus with just 4 genes. Most essentially, one coding for an essential protein, the nuclear one directing the coding of a nuclear material, one directing the assembly of the virus, and the other one directing the excision of the virus from this host cell. That's the simplest form we can see. In some cases, they might enlarge to include a membrane envelope. So some viruses might have additional protectives, a membrane envelope. So these ones will generally be resistant to desiccation and so on. You must be familiar with this virus. I intentionally brought it back, although it's not of great interest as we're growing up. But I brought it back, the T phage, a T even phage. I used to drawing this in your books. You were used to drawing it in secondary school. Yes. Any problem? You were used to drawing this in the secondary school, so I intentionally brought it back so that you'll be able to have an idea of relate what I'm referring to as nuclear material, a protein coat, and a lipid envelope, protective envelope. So with this is the simplest virus that is known, and it infects a bacteria. So we're calling it a phage. A virus that infects a bacteria is called a phage. So again, you see the nuclear material there and a protein shield, and then it has some tail fibers. which are very important, which are very important for infectivity. So it uses these tails to thrust itself. It uses these tails to thrust itself into the bacteria or host cell, and typically trusting its nuclear material. It's a DNA virus, so it trusts its DNA into the host cell.
Someone use Skype.
So it thrusts itself into the host cell and initiate the replication process, which I'm going to describe shortly, but vividly. After it entrusts itself to the host cell, it directs the host cell to synthesize viral DNA alongside viral protons, and the viral protons are assembled. So the viral DNA is packaged into the viral protons, and then it initiates an excision process that ejects the viral particle out of the cell. So this could progress through a lysogenic fashion or a lytic fashion. So why is this important to us? The manner in which a virus infects a cell determines whether the host cell dies or survives after this infection. Okay, I'm just checking my messages on Skype because that is where my recording is, to ensure that the Skype line is very perfect. So I was saying that when a virus infects a cell, the manner in which it progresses to replicate its protons and its nuclear material could be via a lytic fashion or a lysogenic fashion. We'll use phages to exemplify this concept. We use phages to exemplify this concept, and most phages are DNA viruses. I'm going to talk a little bit on how do we classify virus as a DNA or RNA shortly. But in the lytic phase, the phage infects the host. After infecting the host, it takes control of the host machinery and directs it to synthesize viral proteins and viral nuclear material. Directs the assembly of viral particles, and then initiates a puncture on the cell membrane of the host. When that puncture is achieved, the cell becomes susceptible to osmotic shock, is filled with fluid, and then bursts off, releasing the viral particles, usually 100 or more. So these newly released viral particles are now able to infect another virus and initiate a circle all over. So I'm saying that this viral particle thrusts its nuclear material into this E. coli cell, directs the E. coli cell to synthesize viral genome and viral protons. The viral protons are assembled, and upon assembly, it initiates a process that produces a porous puncture on the cell membrane of the host, and therefore it becomes susceptible to osmotic shock. It's filled with fluid, and then the cell bursts. So at the end, at the end, the cell dies, while the viral particles are able to initiate another cycle of infection. Bacteria have developed a lot of means to resist this. Some are generally resistant. Mutant strains are resistant to viral infectivity. Some have even developed a restriction nucleases to cut and chop off viral DNA when it is thrusted into the bacterial cell. These are just highlights of protective mechanism by the bacteria, not essential for this course. Now, in the lytic circle, which is different from the in the lysogenic circle, which is different from the lytic circle I just explained, the viral particle does not kill the host. That is the first hallmark to note on the lytic cycle. The viral particle does not kill the host. So the host survives after the viral replication. Now, how does that progress? That is what we're going to note here. First, the viral particle entrusts or thrust its own DNA into the host cell instead of the viral DNA taking hostage the host cell genome and directing the synthesis of viral particles and proteins, and nuclear material. The host DNA integrates itself into the viral DNA. The host DNA integrates itself into, the viral DNA integrates itself into the host DNA, forming what we call a recombinant. So the host DNA is now a recombinant DNA molecule of one, the viral DNA, and two, the host DNA. Could be a bacteria, so the bacterial DNA. So as the host DNA replicates in an attempt to replicate itself, it also replicates the viral DNA strand that is incorporated into its own DNA. So if it replicates itself 100 times, the viral DNA would have been replicated 100 times as well. The viral DNA then directs the synthesis of viral proteins and other viral particles, and also directs the assembly of the viral particles, and then by some means it finds itself out of the cell. I missed a step here to point out that before the viral DNA directs itself, it has to somehow disintegrate itself from the host DNA. Okay. So what I'm trying to explain is that just a moment. This is the host. This is the host DNA, let's put it like that. So when the viral particle infects the host, we are going to have a new DNA, which is a recombinant. We're going to have a new DNA, which is a recombinant. So this becomes a new DNA after infection. So this portion is the viral DNA, which has been incorporated or integrated into the host. So as the host replicates itself, as the host replicates itself, as the host makes copies of itself, it also copies along the viral DNA. also copies along the viral DNA. So if the host replicates itself 100 times, we are going to have 100 copies of the viral DNA. So at some point, there's a disintegration of this portion. At some point, there's a disintegration of this portion, and this disintegration results in this directing the synthesis of viral particles, which subsequently leave the cell. At one point, a lysogenic phage becomes lytic. At one point, a lysogenic phage becomes lytic. So upon this disintegration, it now progresses as a lytic virus. By doing what? Directing the perforation of the host cell membranes, and of course, the infiltration of fluid, resulting in osmotic imbalance and the bursting of the cell. So the manner in which these viral particles leave the cell could maintain lysogenic properties or could switch to a lytic cycle. So the main summary I'm trying to bring out here is that there's no clear virus that typically replicates in a lysogenic fashion. Occasionally, a viral particle exits the cell and initiates a lytic cycle. But when it remains integrated in the cell, it progresses in a lysogenic fashion. I want to know if that is clear. Okay, good. That is it about replication of viruses, lysogeny or lytic fashion. So let's try to classify viruses. Viruses are generally classified based on the type of nuclear material they contain, they possess. Remember, the first principle of viruses is that they have either DNA or RNA, and never both. So a viral particle has either DNA or RNA. It is never both RNA or DNA. So within this DNA or RNA, there are further classifications of the virus. So a virus could have a double-stranded DNA. An example is the human papilloma virus that causes the cervical cancer, which is a major issue in the world today. An example again could be the adenoviruses, usually respiratory viruses that could cause tumor as well, and the herpes simplex type 1 viruses that usually cause this sores, and type 2 that will make cold sores around your lips, and type 2 that will cause genital sores and shingles and so on. The poxoviruses that causes smallpox and cowpox are also classified in this range. So they are all double-stranded DNA viruses. Also, it could be a single-stranded DNA virus. It could be a single-stranded DNA virus. So you also find the parvoviruses here classified on this, but remember that these are very rare. These are very rare situations or viruses. The third classification is a viral particle having a double-stranded RNA. A viral particle having a double-stranded RNA. An example is the reovirus that causes diarrhea and other respiratory diseases. Then we have a viral particle having a single-stranded DNA. Here we have three other classifications. The first classification is a single-stranded DNA, sorry, a single-stranded RNA that can serve as a messenger RNA. So what are we saying here? That when the virus infects you, the nuclear material of the virus directly starts serving as what? A messenger RNA for the synthesis of what? Viral proteins. I'm sure you must have heard of messenger RNA vaccines that are currently being used now in COVID-19 prevention. And if you have been reading some scientific literature, you realize that it is the very first of its kind. This is the first time a messenger RNA virus or vaccine is being used across humanity. It is a technology that has been under development for some time now. It's not as if they started as COVID started, but the experimentation and the application is the very first of its kind. Of course, scientists are very keen to observing the progress of that. So the second classification of single-stranded RNA viruses is a virus that has its single-stranded RNA serving as a template for the synthesis of a messenger RNA. So in this category, the viral RNA does not serve directly as a messenger RNA, but it serves as a template to use the host enzymatic machinery to synthesize a messenger RNA from it. So again, you have examples here. The rabies virus, measles virus, mumps virus, influenza virus, and other automyxo viruses. And the third classification is a single-stranded RNA viruses. Is a single-stranded RNA virus that uses its own nuclear material to serve as a template for the synthesis of a DNA. So this virus, when it infects a host cell, it uses its own nuclear material, which is a single-stranded RNA, to direct the host cell to synthesize a DNA from it. So this DNA synthesis, that is what we call reverse transcription with the use of the enzyme reverse transcriptase. This newly synthesized DNA now directs the synthesis of a messenger RNA and then viral proteins. So a typical example is the HIV virus. I'm sure you must have heard of a retrovirus before. So this uses a reverse transcriptase enzyme to reversely transcribe its own RNA to a DNA and then DNA back to messenger RNA and proteins. So this is just to bring you the basic classification of viruses, but there are other methods that are used to classify viruses. Again, we could use the host range, we could use the disease range, and so on to classify viruses. But this is the backbone for viral classification based on the type of nuclear material found inside the virus. Okay. After looking at the classification, let's look at isolation, cultivation, and dictation. We isolate viruses because we want to find out if the virus is actually present, and from there we could use it to either prepare solutions for vaccines. We could use it to do further research to understand the nature of the virus. We could use it to perform so many different experiments in the laboratory and study the viruses much better. Viruses, unlike all other particles that have the means to anchor themselves, actually anchored to surfaces by a combination of hydrophobic and electrostatic forces. So they rely but on a combination of hydrophobic and hydrostatic forces to anchor themselves onto substratum. So this could be surfaces of material, your hands, or food items, liquid, and stuffs. So a viral particle could be anchored on your hand simply because of the surface tension on your hand, meaning that if that surface tension is reversed, the viral particle is going to fall off. So for instance, if you apply a detergent on your body, a detergent is capable of reversing surface tension, and therefore that virus is likely going to fall off. That is why hand washing has been very prioritized in the control of COVID. So to isolate a virus, therefore, we take a sample that we're supposed to contain the virus. We'll try to obstruct the current balance of the sample. For instance, we could add beef extract onto the sample. The addition of beef extract is going to upset the pH of the sample, and therefore, if the virus was anchoring within an acidic medium, it is going to dissolve because of the alteration or because of the increase in pH. So when we alter the virus from the original surface into our own surface, we can now filter our own surface or filter the original surface. And now we have a filtrate that is either rid of the virus or contains the virus, depending on which one we used. But the typical proceeding is that we introduce the beef extract to dissolve the viruses from the original substrate onto the beef extract. And then later, we filter off the beef extract, which now contains our viruses. Okay, we could again rebalance the pH because the virus was surviving at that pH. If we maintain a different pH, we might kill the viral particle. So it's important to restore or reinstate the pH balance so that the viral particle is still viable for further studies, especially culture-based studies. And when we filter using a filter that is capable of capturing all particles except our viral particles, and therefore we are able to move on and get it well prepared. And upon obtaining this filtrate that contains our viral particle, we could progress to do further studies on the virus, as I indicated, maybe for vaccines, for inoculation of susceptible animal hosts, for further experimentation, etc., etc. So the Zoom link is going to restart in less than a minute. When we come back, we'll look at cultivation of viruses. Of course, the rule remains the same. Immediately the link expires, you just redirect the link, you'll be back in class. Now, for the students on Skype, I hope the screen was constantly shared throughout.
Yes, so the screen was showing. We were seeing everything.
Okay, good. We are going to wrap up within a short time, then we move on to plenary discussion. As I said, it's very important to have the Skype screen shared because that is where I am able to capture recordings. So Skype permits me to record continuously. If I use Zoom, for instance, after this break interruption, it will mean that I'll have to restart. And just to note that this is the last class we're having a combined lecture. After that, we'll concentrate only on our section. I combined these two sessions because they were overlapping with another course I'm teaching. But subsequently, we'll have only our Skype class, and there will be no Zoom addition. And we'll move on, with a lot of serenity. So I think this is the second and last class that we'll be having this combination. I'm talking to the guys on Skype. I hope that is okay.
Yes, sir. Yes, sir.
The Zoom class is on back. Let's go on so that we don't have delay with recordings. Okay, welcome back, the Zoom people. First confirm to me that you can see my screen. That's good. Skype, you can still see, right?
So it has disappeared though. We're no
You see now?
longer see.
Okay, so
Still blank.
let me reshare it. Let me reshare it. Skype, can you see again?
Yes, sir, I see.
Okay, good. Let's
No,
go
sir.
on with the cultivation of viruses without further delay. We have isolated the viruses. So I'm saying that we now have a certain liquid medium, which we assume that contains, remember, we can't see them as we are doing all this manipulation. We are unable to see them. So we have a certain liquid medium, which we assume contains our viruses. And that is the basis on which we are going to work. Okay. So we have filtered off. This is a certain filtrate that we have. So as you can see, you have some bulbs in this filtrate. We are assuming that these bulbs are our viruses. And therefore, we can go further now to culture the viruses. So as I indicated earlier, we are unable to culture viruses using synthetic media. We are able to culture viruses using synthetic media because they don't have the capacity for independent replication. They are also unable to synthesize their own proteins. So we will always require a living host, a living tissue to synthesize, to culture these viruses. So there are different types of living tissues that we use for this purpose. We could use bacteria, but this is only if the virus is capable of infecting a bacteria. So we use bacteria only for what? Bacteriophages. If it's not a bacteriophage, there's no way we can use a bacteria. But we could also go further to use egg embryo. Remember, an egg embryo is a living tissue. So some viruses are capable of growing in an egg embryo. So we can use an egg embryo to culture the viruses. And in this case, we have to be very careful where we inoculate, because viruses, as I indicated in my very first lecture, are very specific to their host. And even within the host, some viruses are very specific to tissue. So there are some that will infect only the charioalveolar membrane. There are some that will infect the amniotic sac cavity. There are some that will infect the embryo. There are some that will infect by the yolk sac. So you must know the tropism of the virus tissue before you can move further to inoculate. For animal viruses, we typically use an animal. So we use animal host, or we use tissue of the animal. So for animal host, we typically use small animals like mice, small laboratory animals like mice for this purpose. But for viruses that want to really ascertain their infectivity in human host, we could use human tissues like the HeLa cells. So with this, we will note two types of human tissues: primary cell culture and continuous cell culture. So a primary cell culture, we simply harvest tissue cells from a tissue. It could be a human tissue, a kidney tissue, or liver cells, either from a human, a monkey, and stuff. So when we harvest these tissues, we put them on a plate culture, and then we infect them with a viral solution. When we infect them with a viral solution, if the virus is able to infect these cells will not grow. Or there are signs on the cell, there are changes on the cell that will be signs of viral infectivity. I'm going to talk about these changes shortly. We typically call them cytopathic effects. But we need to observe that our cells are living cells. So to observe that, we culture them without infection yet. And we know that primary cells don't grow when they meet each other. I'm sure you must have heard of the phenomenon of contact inhibition before. So when cells grow and get in contact with each other, they stop growing. There's a method of growth control even right in the system. So when a cell grows and meet each other, they stop growing. This is contact inhibition. And if we want these cells to grow further, what do? We reduce. So we partition them so that they'll grow further. But our essence is not to grow cells. Our essence is to have cells on which we can infect and observe for viral infectivity. The next thing to note about this primary cell culture is that just like your own cells, since they are harvested directly from the living tissues, they cannot divide infinitely. So at one point, you will have to stop dividing. That is why at one point you will have to die. Whether you eat the best food on earth and you live the best life on earth, at one point you have to die, because at one point your cells stop dividing. They have divided to the finite division. And when they stop dividing, you have to die. So that is typical too of primary cells or primary cell lines. At one point, they'll stop dividing. Now, I describe it like you go and harvest liver cells, you go and harvest kidney cells and so on. It's not a straightforward stuff like you kill a cow and harvest the cells and you go to a laboratory and you're really infecting with viruses. It is a very sophisticated proceeding, very expensive, and very resource demanding, both in terms of human resources and material resources. So we cannot afford to go and harvest anytime we want to culture a virus. We don't have the luxury of going to harvest the cells anytime we feel like cultivating a virus. So what are we saying? We have to be able to maintain this culture for infinite uses. We have to be able to maintain these cells for infinite uses. First, they have a limitation. They are substitute to contact inhibition, and then second, at one point you have to stop dividing. So to bridge this gap, scientists have been able to transform primary into what we call cell lines. Scientists have been able to transform these cells into what we call continuous cell lines. You see the background under which some of you attend classes. We are waiting. Okay, so I'm showing scientists have been able to transform these cells into what we call continuous cell lines. And what is the transformation here? The first transformation is that they have been made to lost their phenomenon of contact inhibition. This is common. Excuse me. This is common with cancerous cells. Cancerous cells are cells of the body that have lost this phenomenon, and they'll grow on top of each other. So they'll grow on top of each other, grow on top of each other. That's why you see the outgrowth and tumorous exhibition. The other situation is that these cells have been transformed to divide infinitely. So they can divide unrestrictedly. Again, this is a phenomenon common in cancerous cells. A cancerous cells will divide and divide and divide and divide. Nothing stops us from dividing. So normal human cells are transformed to lost this phenomenon of contact inhibition and also to enable them divide infinitely. So if you look at what I just said, I'm describing a cancerous cell. So some of the cell lines that are being used in scientific investigation today are cancerous cells. The HeLa cell line, for instance, were harvested from the cervix of Henrietta Lacks, who died of cervical cancer. So scientists have confirmed that she had cervical cancer, and just as after she died, cells from her cervix were harvested more than 50 years ago, and up to today, these cells are serving scientists. I said 50 years ago, I think more than 70 years ago. These cells are serving scientists across the world to investigate the growth of viruses and for vaccines and so on. So again, you can read the story of Henrietta Lacks, and the world has come recently to acknowledge her contribution to the scientific world, and there's a lot of retribution being done there. So I've identified the different media on which we can culture viruses. I started by saying there are bacteria. We can culture on eggs. We can culture of small mammals. We can culture on cell lines. It could be primary cell line. It could be continuous cell line. Good. So after cultivating, we need to be able to identify if indeed there was growth. We need to be able to identify if there was growth. So on bacterial culture, we prepare a bacterial colony, just as we do in the laboratory. So we prepare our agar. The agar is usually prepared to be more soft. So we have 70%, the media, is prepared to be more soft. So we have a lesser concentration of agar there than what we typically use in bacterial growth. So we prepare our agar, and we allow the bacteria, we inoculate the bacterial culture using the spread plate technique. A spread plate technique is a technique we'll also be learning during our practicals. So we inoculate our bacteria using the spread plate technique, and then we infect the bacterial lawn with our viral solution. The assumption here is that where a single viral particle exists and there's a bacterial cell, the viral particle would infect the bacterial cell. And the viral particle, if it's a lytic phage, will break down the bacterial cell, and there will be no growth around where there was infectivity. So following incubation for 24 hours, if we inspect or observed for area of lack of viral growth. This is a lawn. Normally, if there was no infectivity, we are going to see but a lawn that has grown. If you look at this picture here, this picture I'm pointing here, you see that this area, all of this area is a lawn of bacterial growth. Okay. Now you see some small patches. These are called plaques. These patches are called plaques. If you see here, there are plaques. If you see here, there are plaques. So here there are more plaques, there are fewer plaques, and there are much fewer plaques. Now, as I said, this plaque is representing an area where a virus infected a bacteria and the bacteria could not grow. So we have a clear zone there. So if we count this number of clear zones, we can be able to tell you how many viral particles were in our solution by expanding it now to the volume of the inoculum. So you see here that there were more viral particles here and than here. So we can quantify the viral concentration by counting these plaques. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So we'll say there are 14 plaques forming units per meal if it was 1 ml of the solution that we applied here. So it is expressed as a function of the volume of the viral solution. So first, we are able to identify that this virus has grown. This virus exists and it has grown by infecting this bacteria. And then we can also quantify the number of viral particles that were in our solution. For egg embryo, we infect, as I indicated, we inoculate the virus into a specific region on our egg. Inoculate the virus into a specific region on our egg, and we can observe for signs of viral infectivity. Observe for signs of viral infectivity. These signs could include what? These signs could include death of the embryo, defects with growth of the embryo, could observe damaged areas on the membranes of the embryo, and this damaged area could result in discrete opaque patches on the embryo, and these discrete opaque patches are called pucks. Discrete opaque patches on the embryo called pucks. So take note, viral infectivity could result in death of the embryo, developmental defects, localized damages on the embryo, or these discrete opaque patches that you see on the embryo. For animals, small mammals that we infect, signs of viral infectivity could also include death of the mammal, growth defects, or abnormal development of the mammal. For cell lines, we typically observe signs of cytopathic effects. sites of distortion in the typical presentation of a cell line. And this could include loss of adherence of the cells to the substratum. So all cells are anchorage dependent. When they grow, they anchor themselves to the substratum of the container or the medium. So when they lost this ability, it's an indication of infectivity. The shape of the cells to become could become a normal flat cell, becomes a rounded cell. The nucleus could shrink in size and could have so many vacuoles present in the cytoplasm. The cytoplasmic membrane could fuse with the nucleus, forming what we call a multi-integrated synthetia. We could also have inclusion bodies in the nucleus, and in worst cases, we will have complete lysis of the cell. That is for a lytic cycle. So take note of all these signs of cytopathic effects, which are indication of infectivity. There are other situations where one able to detect these viruses using all these techniques I've just mentioned. We are able to identify them on bacterial colony because they are not bacteriophages. We are unable to identify them on embryo because they don't infect egg embryo. We are able to identify them in culture because they don't produce visible signs of cytopathic effects. Therefore, we might have to go to use other concentrated acids. We might decide to use other concentrated acids. For instance, some of these viruses could agglutinate red blood cells. So we'll be able to use heme agglutination assay to observe for signs of viral infectivity. The typical model used is what we call heme agglutination inhibition assay. We will also exploit the ability of viral particles, specifically antigens, to react with antibodies in what we call immunological assays. There are different types of immunological assays, but the most commonly used is ELISA, enzyme-linked immunoglobulin assay. And finally, we could amplify nuclear material, the nuclear material of the virus, and dictate the presence of this nuclear material. I'm rushing over this because I don't expect you to be able to master all of it, but it is detailed in the notes to a certain extent for your appreciation. So you have the notes there, and I'll be identifying your specific groups who has to take particular interest in what. I'm still going to meet you guys in your specific groups. That's why I've just highlighted this. For some groups, this highlight is okay, but for other groups, you need greater detail, and I'll be meeting you in your separate forums to expatiate on this further identification. If we cannot, how do we explore the ability of the virus to absorb and agglutinate red blood cells, ability of the virus to react with serological components, antibodies, and our ability to use amplification techniques to detect the viruses. So I'll be expatiating on this when it is necessary. So with that, I say we have come to the end of the lecture for today, for the part 1, of course. So I will stop the recording.